In the earlier class, we studied about double fertilization. In this class, today we will see what changes have taken place in the embryo sac and the surrounding regions of the ovary after double fertilization. So let's see the diagram first. One of the male gamete, it fuses with the egg cell and forms the zygote. The synergids that are present here will disintegrate slowly. In the chalaza side, the antipodal cells will also disintegrate. One male gamete, that is the second male gamete, fused with the two polar nuclei and formed PEN, that is primary endosperm nucleus, which gave rise to primary endosperm cell, and after further division, the primary endospermic cell gave rise to endosperm. Now, this is one of the major events in the post-fertilization part. The zygote here is formed, is diploid. The endosperm is triploid, 3N. This is also triploid, 3N, and this is also triploid, 3N. The zygote will develop into embryo. So development of embryo is also an important post-fertilization part. The formation of the embryo and the development of the embryo is one of the most important post-fertilization event. The nucellus that was present here during all this development, the nucellus, that is nutritive portion, this is the nucellus, the nutritive portion has been used up by the embryo, the zygote and the polar nuclei. In majority of the cases, the whole of the nucellus is used, but in some cases, some of the nucellus is left, that is persistent nucellus is left. So in normal cases, what happens? Whole of the nucellus is used, but in some cases, persistent nucellus is left. Is present. So that is known as perisperm type of endosperm. Endosperm. So example of this perisperm is, example we can take of black pepper, and beetroot. After double fertilization has taken place, the major events that take part are formation of endosperm, formation of embryo, formation of fruit and formation of seed. These are the major post-fertilization changes that take place. Now we come to the post-fertilization event. So post-fertilization, the endosperm develops first because it has to give nutrition to the growing embryo. So, seeing the nature of the formation of endosperm, endosperm is of three types. So, there are three types of endosperm. The first type is known as free nuclear type of endosperm. Free nuclear type. So this is the first type. In free nuclear type of endosperm, the primary endosperm nucleus undergoes division, mitotic division, that is only karyokinesis takes place. Cytokinesis is not followed by karyokinesis. So primary endosperm nucleus will divide mitotically and form many cells. All are triploid in nature. The nucleus undergoes mitotic division, and forms many nuclei. These nuclei now gets arranged near the periphery of the cell. Best example is coconut. The coconut water, the tender coconut water or the green coconut that we take, is the watery part is actually made up of free nuclei endosperm. Coconut water is the example of free nuclear endosperm. Free nuclear endosperm. The second division is the cellular division. This is found in cellular type of endosperm. So second type of endosperm is cellular type of endosperm. Cellular type of endosperm. In this, the nuclear division is followed by cytokinesis. That is, karyokinesis is followed by cytokinesis. First, the nucleus divides and then the cytoplasm also divides. This can also be seen in the coconut. The free nuclear portion, nuclear division of the endosperm, we saw in the watery portion of the coconut. Now, cellular type of endosperm division is seen in the periphery, that is, the cells will divide, nucleus along with the cytoplasm will divide and form a layer of cell. So, this gives rise to the solid portion of the coconut, the edible kernel of the coconut. Now let's see how coconut is formed. The free nuclear endosperm gives rise to the watery portion. That means the PEC, that is primary endospermic cell, divides mitotically. The nucleus divides mitotically and forms many nuclei, which gives the rise to the watery portion of the coconut. After this, the cellular type of endosperm is formed. That is, the nucleus, division of the nucleus is followed by the division of the cytoplasm. which gives rise to the white kernel, the edible portion of the coconut. This is the edible portion of the coconut. The third type is the heliobiyal type of endosperm. This is an intermediate form between the free nuclear form and the cellular form. The nucleus divides mitotically and then the subportion of the cytoplasm also divides. This can be seen in all types of monocots. So the third type of endosperm division is the heliobiyal type of endosperm. Example is monocot. It's found in many monocot seeds. We have seen three types of endosperm, that is free nuclear form of endosperm. Then we have seen the cellular type of endosperm and the heliobiyal type of endosperm. During the formation of the embryo, whole of the endosperm is used. So, most of the seeds are without endosperm. So, you can call them as non-endospermic seeds. But some of the endosperm is persisted in some cases. That is known as endospermic seed. In the post-fertilization events, many changes take place. The whole of the ovary is converted into fruit. The embryo sac gets converted into ovule. The two integuments, they get converted into seed coat. This we will learn afterwards. After the development of the endosperm, the embryo starts developing. This whole process is known as embryogenesis, and this we will talk in the next class.